Welcome to Study Loop.
This question asks us how the sequences for 2 and 3 are different from the sequence for 1. Well, if we look at 2 and 3, the difference between 2 and 3 is that in 2 we have a UUA, and 3 we have a UAA. The rest of the sequence is the same, right? So UUA codes for leucine. In 3, the UAA codes for a stop codon. How does this differ from 1? Well, 1 has a UCG to start with, which is a serine, rather than a UCU, which is a serine. Interesting. So the first UCG on 1, and then in 2 and 3, we have also a serine, but it's a UCU. Let's look at the rest of it. Well, the rest of them are the same as 2. So is there a difference between 1 and 2 in terms of the peptide that you're going to produce from the mRNA? No. Is there a difference between 2 and 3 in the peptide? Oh yeah. The difference is that 3 is going to be prematurely terminated by this stop on the second to last codon. So therefore, let's look at our choices. Which one's the answer? There isn't a mutation in peptide 2 and 3. Or there isn't a mutation in peptide 2 and peptide 3 has a stop codon. Well, it looks like there is a mutation in peptide 2, but peptide 3 does have a stop codon. There's a different amino acid in peptide 2. Nope. Both serines. Peptide 3 has a stop codon. Yep. There's a silent mutation in peptide 3, and peptide 2 has a stop codon. Nope. Peptide 3 doesn't have a silent mutation. It becomes a stop codon. And peptide 2 doesn't have a stop codon there. Then going to answer choice A, there's a silent mutation in peptide 2. Yep. Silent mutation meaning that the actual amino acid produced doesn't change, but the sequence of nucleotides is different. Okay, so that's true. Peptide 3 has a stop codon, and that's also true. So our answer choice A.